I think I gotta tighten my wire back there or something. Try that again. But one of the things I wanted to get out on this ride was a shout out to the Alex Reyes paper that I mentioned not so briefly in my first journal club on this channel. So I put the links in the link in the description below to that paper. But Alex Reyes, this first author paper by him, and out of Bert Sackman's lab with Nail Bernyshev and Andrei Razov, they do a pretty extraordinary thing, especially in the context of what was going on at that time. This whole patch clamp methodology was already considered pretty cutting edge at this time in the early 90s, and then not very many labs are doing it, and they just recently got the Nobel Prize for doing it, and so everybody wants to learn to do it, but you need to go and train somewhere. But in this paper, just a quick synopsis, it's one of the first papers that really takes paired recordings and makes an incredibly cool observation. And this is, not too long after. It's like the field was changing at this time. They gave away the Nobel Prize or awarded the Nobel Prize, I should say. That was almost like a slip there. Bert Sackman and Erwin Nair were awarded the Nobel Prize in, I think, 1991 for single channel recordings. But coming along with that discovery is just the whole methodology of patch clamp, and wholesale recordings came along with that too, and we've just gained so much of our knowledge about how individual neurons work from this technique, or this combination of technique, this family of techniques. So Sackman and Nair's contribution to science in general, and neuroscience and biology as a whole, as a subfield of science, because it contributed to cell biology and neurobiology and cardiology, and any and everything has benefited from electrophysiological recordings from. So a big shout out to the Alex Reyes paper. I don't know if this was it's one of his first papers, and I'm sure it remains one of his most important papers just because it is a seminal discovery in neuroscience. In this paper, he describes the phenomenon that a single pyramidal cell can make synapses with different short-term dynamics depending on the target cell. And I think that's an extraordinary thing. If you read that abstract, it's just awesome, because every sentence is gold. And they speculate that it's a presynaptic mechanism. They show that it's also that interneurons can make synapses with differing short-term synaptic dynamics. And so then they understand that the way that inhibition will be evoked in the local microcircuit when a pyramidal cell fires is dependent on the frequency that the pyramidal cell fires at. They make that jump already cognitively, in their imagination. They're able to make that jump. No model necessary, because it's obvious, right? But that's the state that neuroscience was in. We knew so little about short-term synaptic dynamics. We made so many assumptions. There were so many unanswered questions. And so when you make this paired recording in the cortex for the first time, you're guaranteed to find cool stuff. happy thing is that they were clever enough to design experiments could tease out some obvious functionality like short-term synaptic dynamics. To notice that, I think up until now, up until this time, paired pulse, that's just two action potentials, was a sort of a thing. So you don't get much dynamics from that. You do to a certain extent, right? But it's a ratio. Whereas if you have a train of action potentials firing and driving the synapse, then you can see how the synapse would react to a burst of activity or to tonic activity. And so that's what makes this paper so freaking cool, is that this is a patch clamp paper from the 90s. We're in 2019 now. And so a patch clamp paper from the 90s was already doing something so amazing. And that's what I still see as the utility of patch clamp, provided that you have a sort of a theoretical understanding of what action potentials mean in a particular brain area. Then I think it's very, very useful to add to that theory concrete evidence about how the microcircuit maps.